Let us now study about general or homologous recombination. General recombination is an integral part of the complex process of meiosis in sexually reproducing organisms. It results in a crossing over between pairs of genes along a chromosome, which are revealed in appropriate matings. The chiasmata that link homologous chromosomes during meiosis are the likely sites of the crossovers that result in recombination. As you can see in this figure, the homologous recombination occurs between similar but not identical copies of the human chromosome 1 during meiosis, which produces new combination of genes. General recombination also occurs in non-sexual organisms when two copies of a chromosome are present. Examples are recombination during F factor mediated conjugal transfer of parts of chromosomes in E. coli, recombination between two phage during a mixed infection of bacteria. As you can see in the following figures, the F factor mediated conjugal transfer of parts of chromosomes in E. coli can be seen. In the following figure, recombination between two phage during a mixed infection of bacteria. The nucleotide sequences are exchanged between two identical molecules of DNA in homologous combination. The most important application of homologous recombination is double strand break repair. During the process of meiosis, homologous recombination produces new combinations and results variation in offspring, which in turn enables populations to adapt during the course of evolution. Exchange of genetic material between different strains of bacteria and viruses through horizontal gene transfer can be used in gene targeting, a technique for introducing genetic changes into target organisms. Let us now study about the different models of homologous recombination. Currently, two main pathways have been proposed how homologous recombination repairs DNA lesions. The double strand break repair or DSBR pathway is used to describe recombination in meiotic cells, predominantly creating crossover products that ensures the accurate segregation of homologs. The synthesis dependent strands annealing or SDSA pathway, on the contrary, typically generates non-crossovers and describes mitotic DNA repair. Homologous recombination repairs DNA in the S and G2 phases of the cell cycle. This is when duplex DNA strands are more easily available and close to each other. Homologous recombination starts with resection of the double strand breakage in the 5' to 3' direction by the MRE11 Rad50 NBS1 complex, forming a 3' single stranded DNA fragment to which Rad52 binds. Rad52 interacts with Rad51, provoking a DNA strand exchange with an undamaged homologous DNA molecule. Assembly of the Rad51 nucleoprotein filament is facilitated by different Rad51 and BRCA2. The single stranded DNA is attached to a ring complex formed by Rad52. This protects against exonucleolytic digestion. Rad52 gene in humans synthesize Rad52 protein, which interacts with Rad51 and replication protein A or RPA and activates Rad51. In eukaryotic cells, RPA binds to single stranded DNA and then binds the 3' overhangs. RPA prevents single stranded DNA from winding back on itself or forming secondary structures during DNA replication. Rad51 and BRCA2 protein forms nucleofilaments, binds single double stranded DNA and interaction with the homologous section on an undamaged DNA molecule called strand invasion. Rad51 will catalyze the strand exchange in a D-loop shape and the DNA synthesis will happen on the invading strand. When this is done, the repaired DNA and the undamaged duplex DNA will change from a D-loop to form a cross-shaped junction, also known as Holliday junction. There are two main models which suggest the pathways the holiday junction DNA will break into two separate strands, SDSA pathway and DSBR pathway. The figure on the screen shows the SDSA pathway with no crossovers. Complementary base pairing anneals the newly synthesized 3' end of the invading strand to the other 3' end overhang in the damaged chromosome. Remaining flaps of DNA are removed. SDSA pathway finishes with the ligation of single-stranded gaps. In the DSBR pathway, the repair process starts by the degradation of the 5' ends of the double-stranded break or DNA resection. The resulting single-strand DNA ends and then invades the homologous DNA sequences, thereby forming heteroduplex DNA and serve as primers for DNA synthesis. After ligation of the newly synthesized DNA to the resected 5' strands, an intermediate containing two holiday junctions is formed. Resolution of this joint molecule and mismatch repair results in crossover or no crossover products depending on how the double holiday junction is cut. The DSBR pathway commonly results in crossover. Chromosomal crossover will occur if one holiday junction is cut on the crossing strand and the other holiday junction is cut on the non-crossing strand. This can be seen as shown in the figure along the horizontal purple arrowheads at one holiday junction and along the vertical orange arrowheads at the other holiday junction. Alternatively, if the two holiday junctions are cut on the crossing strands, then chromosomes without crossovers will be produced. Holiday model. Homologous genetic recombination is essential in maintaining genomic stability such as to protect genomes from double strand breaks or interstrand crosslinks. It is also important in generating genetic diversity that is necessary for evolution. Reciprocal exchange of DNA strands between two nearly identical DNA molecules occurs during homologous recombination, results in a four-way DNA junction named as holiday junction and is named after Robin in 1964. The following figure explains the holiday model of DNA crossover. In the first step, two homologous DNA molecules line up. Next, the cut in one strand of both the DNAs occur. The cut strands cross and join homologous strands forming the holiday structure or holiday junction. Next, heteroduplex region is formed by branch migration. Resolution of the holiday structure. DNA strands may be cut along either the vertical line or the horizontal line. The vertical cut will result in crossover between F, F prime and capital F, F prime regions. The heteroduplex region will eventually be corrected by mismatch repair. Finally, the horizontal cut does not lead to crossover after mismatch repair. However, it could cause gene conversion. The holiday junction is not a static structure. It can undergo spontaneous branch migration in which the branch point can hop forward or backward in a stochastic way. Extensive branch migration requires branch migration enzymes to provide a direction over long stretches of DNA. In between of adjacent branch migration events, the holiday junction can spontaneously fold or unfold, transforming from one stacked conformer to the other. The holiday junction can be resolved into two duplex DNA molecules via the action of junction resolving enzymes, and the extent of genetic information exchange depends on the orientation of resolution. T7 endonuclease 1, a junction resolving enzyme, binds to the holiday junction with high specificity and makes two cuts diagonally across the junction. The holiday junction plays a large role in genetic recombination. The different arms allow the junction to be mobile. The main purpose of the model is to explain the mechanism of gene conversion during the recombination event. At the location of reciprocal exchange is a distinct marker. Advantage of recombination. Here in this figure you can see the biological roles of recombination. Cell divisions in eukaryotes like plants, animals, fungi and protists that divide through mitosis, where the integrity is maintained by homologous recombination through double strand breaks. The unrepaired double strand breaks can lead to cancer. Tumor suppressor genes BRCA1 and BRCA2 was to play a key role in regulating recombination in humans. Individuals who are heterozygous for BRCA2 are subject to increased risk for breast and ovarian cancer, and loss of both alleles causes Fanconi's anemia. Rad51 binding to single stranded DNA was found to be promoted by BRCA2. This type of recombination brings out diversity in a population. Let us now study about the various applications of homologous recombination. The various applications include gene targeting, protein engineering and cancer therapy.